In this video tutorial, I am going to discuss about absorption of amino acids and protein. In the previous video, we had seen digestion of protein, and in that video, we had seen that at the end of protein digestion, we are left with amino acids. Some protein digested up to only di or tripeptides, and very little, approximately 5% of the protein, it remains either undigested or partially digested. So, in this video, we have to see that how this end product of protein digestions are getting absorbed through the intestinal cell. There are various mechanisms by which these end products of protein digestion can be absorbed. Out of these various processes, the most common and most abundant mechanism is the sodium dependent active process. Sodium dependent active process. To understand this sodium dependent active process, suppose this is the intestinal cell. On this side we have a lumen, and on this side we have a blood, and this is our intestinal cell. The cell membrane which faces towards the lumen, we call it as a apical cell membrane, whereas rest all cell membrane, it is called as a basolateral cell membrane. In the apical surface of cell membrane, there is one transporter which is called as a solute carrier protein, which is often abbreviated for solute carrier as a SLC. This solute carrier protein is there on the apical side of cell membrane. What this protein does, it transports sodium as well as amino acid on the same direction, that is from the lumen towards the cell. This solute carrier protein, it do not utilize ATP directly, rather it utilizes ATP indirectly. How? So, on the basolateral side of the cell membrane, there is one more transporter protein which is called as a sodium potassium ATPase. This sodium potassium ATPase, it utilizes ATP and sends 3 sodium out of the cell and 2 potassium inside the cell. This sodium potassium ATPase, this transporter, it is continuously working. So, what happens? Inside the cell, there will be very low concentration of the sodium because it is continuously sending sodium molecules outside of the cell. And as sodium concentration became low in the cell because of this transporter, this solute carrier protein, it can transport sodium towards the concentration gradient, right, from the luminal side to the cell. And along with the sodium, amino acids are also dragged inside the cell. So, we can say that solute carrier protein is indirectly utilizing ATP through this sodium potassium ATPase. And when any carrier protein indirectly utilizes ATP, we call them as a secondary active transport. So, solute carrier protein, it is a type of secondary active transport mechanism, right? And if you think carefully, if you remember carefully, this such secondary active transport, it is also seen in the glucose absorption from the intestine, right? In the glucose absorption, there is a sodium glucose transporter which is utilizing similar looking mechanism, and it is again example of secondary active transport. Remember, this solute carrier protein, it is not only present in the intestinal epithelial cell, but it is also present in the renal tubular cell. So, it is also present in the kidney. Kidney at which place? In the renal tubular epithelium. Okay, and at that also it carries out the same mechanism for the reabsorption of amino acid from the kidney. Now, for the each set of amino acid, there are different solute carrier protein. Till now, approximately 7 different solute carrier protein has been found, and out of the 7 different solute carrier protein, 4 solute carrier proteins are well characterized. So, we will discuss about these 4 different solute carrier protein which are well characterized one. The first one of the first type of solute carrier protein, it transports neutral amino acid. Neutral amino acid which do not have any charges. For example, it transports alanine, it transports glycine, it transports tryptophan, and all the different neutral amino acids are being transported by this. The second type of solute carrier protein, it transports arginine, lysine, cysteine and ornithine. This ornithine is the non-standard amino acid, right? This arginine and lysine both are basic amino acids. Cysteine is a sulfur containing amino acid. As you can see, majority of the amino acid transported by the second type of solute carrier protein is a basic amino acid. So, we call it as a solute carrier protein specific for basic amino acid. It has given a second name also by taking this first alphabet of all these 4 amino acids. So, C, O, A and L. So, it is also called as a coal transporter. Remember, the solute carrier protein, they are present at both the places, intestine as well as kidney. The third type of solute carrier protein, it transports acidic amino acids. So, specific for acidic amino acid, and these are aspartic acid and glutamic acid. And fourth type of solute carrier protein, it transports proline, hydroxyproline and glycine, right? And as we can see that proline and hydroxyproline both are amino acids. So, this is specific for amino acids. Now, as you can see that glycine is transported by this amino acid transporter as well as neutral amino acid transporter. So, any single amino acid can be transported by multiple transport mechanism, and each transporter has a multiple amino acid. So, set of amino acid is there. Now, what happens, sometime there are a genetic defect by which any of this transporter can be defective. So, many such diseases are there. We will consider some of the disease of this transporter defect. So, whenever there is a neutral amino acid transporter defect, it leads to Hartnup disease. Hartnup disease. So, in the Hartnup disease, what will happen? There will be malabsorption of all these neutral amino acids, right? And as we had discussed earlier, that these transporters are not only present in the intestine, they are also present in the kidney. So, because this transporter is also defective in the kidney, these amino acids will not be reabsorbed through the kidney. So, this amino acid will be lost in the urine. Neutral amino acids will be lost in the urine. Now, most of this neutral amino acid, they are non-essential. So, as such, we don't have any problem. But with the tryptophan, there is one particular problem, because tryptophan, it gives rise to synthesis of niacin. Niacin can be synthesized from the tryptophan, right? And niacin is one of the major vitamin. So, what will happen in such patient of Hartnup disease? There is a deficiency of the niacin which leads to pellagra. Pellagra is nothing but the niacin deficiency, and in the pellagra we can see diarrhea, dermatitis, dementia, all these conditions can be seen. So, this can be a major problem because of loss of tryptophan. Second, that if basic or coal amino acid transporters are defective, then what will happen? Then again all these amino acid from the intestine, they will not be absorbed, and second, through the kidney all these amino acid will be lost. Now, here also generally we do not have any problem, but cysteine, loss of cysteine in the urine creates a problem. It leads to cystinuria. Cystinuria. Now, because of the cystinuria, what will happen? This cysteine is a less soluble. So, there are chances that this cysteine, excessive cysteine in the urine, it will precipitate in the renal tract, and specifically it precipitates at a low pH. So, if there is an acidic urine, then this extra cysteine, it precipitates and gives rise to cysteine stone. So, this will be a pathological condition which require a treatment. There is no any major disease for this acidic amino acid. For this amino acid, it can be defective in certain disease, and this leads to glycinuria. Of course, proline and hydroxyproline and glycine, everything will be present in the urine. They will be malabsorbed through the intestine as well as present in the urine. But classically, it is given a name of a glycinuria. So, remember, in the glycinuria, this particular transporter can be defective. Now, let us understand one more clinical aspect of this solute carrier protein. In the market, now there are availability of the super oral rehydration solution. This oral rehydration solution, they are given in case of the diarrhea. In the diarrhea, what happens? There is a electrolyte and water loss. right? And for the treatment purpose, we provide them with the oral rehydration solution. Now, the classical oral rehydration solutions, they contain sodium and glucose among other things. Why they contain sodium and glucose? Because sodium and glucose, just like this amino acid, they are absorbed together. So, what will happen? Sodium will reabsorb. So, electrolyte imbalance will be corrected, and along with the sodium, water is also reabsorbed. So, this water loss is also corrected. In case of super ORS, along with the sodium and glucose, amino acids are also added. So, super ORS, they are nothing but the ORS which are fortified with the amino acid. So, what will happen? Now, earlier only glucose was symphotic with the sodium. Now, by providing amino acids in the ORS, this more and more sodium will be reabsorbed through the intestine, right? So, the fluid imbalance, fluid and electrolyte imbalance can be rapidly corrected by the super ORS. So, this thing we have to keep in mind. Now, let us discuss about the second important transport mechanism or absorption mechanism for the amino acid. The second mechanism is by the proton linked peptide transporters. Proton linked peptide transporters, and sometimes they are abbreviated as a peptide transporter 1. So, PEPT1. Now, to understand this mechanism, suppose this is the intestinal cell, and on this side we have a lumen, this side blood, and this is the cell. On this apical cell membrane, there is this peptide transporter 1 is present, PEPT1. Now, what this peptide transporter does? It transports dipeptide and tripeptides. And this transport of dipeptide and tripeptide inside the cell is coupled with the transport of this H plus, or we can also call it as a proton, right? So, once this dipeptide and tripeptide, they are transported inside the cell, they immediately hydrolyze to free amino acids. So, there is a hydrolysis, and by this simple mechanism, amino acids, or you can say dipeptide and tripeptide, they are absorbed and immediately converted to amino acid. Now, you might be wondering that what will happen to this amino acid which are inside the cell. Here also there is a amino acid which are inside the cell. So, by whatever mechanism, it may be sodium dependent active process, it may be by this proton link peptide transport 1 mechanism, by whatever mechanism amino acids are absorbed, they are immediately transported through this basolateral portion of cell membrane into the blood by the facilitated diffusion. So, there is one transporter which can transport amino acid, and so it does not utilize any energy. So, we call it as a facilitated diffusion, and by this mechanism amino acid will be transported into the blood, and intestinal system, they are nourished with the portal blood. So, once amino acid reaches to the portal blood, what will happen? They will be transported to the liver, and in the liver most of the amino acids, they are further metabolized. However, there are certain amino acids which are not metabolized in the liver, for example, branched chain amino acids. So, branched chain amino acid, they are not metabolized by the liver, rather liver just transport this branched chain amino acid towards directly to the skeletal muscles. So, this thing also we have to keep in mind while discussing about the absorption of various amino acid. The next mechanism that we are going to discuss is very simple. It is for the transport of D amino acid. Suppose in the diet, if you are taken by mistakenly some D amino acid, then through this intestinal cell, D amino acids, they are directly transported by the passive diffusion. Okay. So, D amino acid can directly cross this apical portion of the cell membrane from the intestinal lumen inside the cell, and there is no any transporter required. It is simply passive or simple diffusion. Passive diffusion. The next mechanism is very important in the neonates or infant. In the infant, what happens? There is a presence of sodium dependent active process, there is a presence of proton linked peptide transporter, but the major mechanism in case of infants is the pinocytosis. This pinocytosis is non-specific mechanism. What happens? Suppose on this side we have intestinal lumen. This is the intestinal cell, right? So, what happens in this infant or neonatal GI system, gastrointestinal system, it is not well developed, and protein digestion is also not complete. So, what happens? There will be various fragments of peptides, and these fragments of peptide, they are directly absorbed through this apical cell membrane. How? So, first there will be creation of such pouch on this apical portion of cell membrane. It will engulf this partially digested protein directly. It is not free amino acid, rather it is a small peptide. Then there will be a pinching off from this cell membrane. So, now the protein particle is inside this vesicle, and this is how these particular proteins are absorbed through the gastrointestinal system. Now, this process has certain advantages and certain disadvantages also. Let us first talk about the advantages. See, infants, they are mostly on the mother's milk, and mother milk, it contains various antibodies or immunoglobulins, right? These antibodies are in the protective nature, for the infants. So, what happens? Antibodies are directly absorbed like this. So, it provides protection to the newborn or infant babies against various pathogens. But it may create a problem also. Suppose other than immunoglobulin, if some antigens are absorbed, what will happen? This antigenic property protein, it will now create a various antibody in the in the infantile system. So, it may create allergic reaction. And similar allergic reaction we can also see in some adults. In some adults also this limited pinocytosis occurs, and if antigenic property protein is ingested, then it may create a allergic response. One of the very good example is the non-tropical sprue. In this non-tropical sprue, what happens? When we eat wheat, any product which are made up from the wheat, so what happens? Wheat contain one protein, that is gluten. When our body tries to digest gluten, it, as the end product of this gluten digestion, there will be 7 to 8 amino acid long oligopeptide. This oligopeptide is antigenic in nature. So, if this oligopeptide, it is absorbed through this like pinocytosis, what will happen? It will create inflammation of intestinal mucosa. Okay. So, this condition is a non-tropical sprue. So, we have to remember advantages as well as disadvantages of this pinocytosis. But remember that pinocytosis is a non-specific one, and mostly it occurs in the infants. Now, the last mechanism that we need to discuss is the Meister cycle, which is also called as a gamma glutamyl cycle. Now, this plays a very minor role in case of absorption of protein. This Meister cycle, it is operative in intestinal as well as kidney. Now, here suppose this is the intestinal cell, on this side there is a lumen, and on this side we have a blood. So, what happens? Whenever there is a free amino acid is there in the lumen, which is the end product of protein digestion, and glutathione inside the cell. Glutathione is nothing but a small peptide. It has 3 amino acid, gamma glutamate, cysteine and glycine. It is a tripeptide basically. Now, what happens? This glutathione which is inside the cell, it reacts with this amino acid in the presence of gamma glutamyl transferase. So, gamma glutamyl transferase, and what happens? This gamma glutamate, it binds with this amino acid, and this cysteine glycine, this dipeptide, it gets separated. So, we get a cysteine and glycine dipeptide, and gamma glutamate, it binds with the amino acid. So, we get a dipeptide having a gamma glutamate and then amino acid, whatever amino acid that we have in the lumen. Later on, this cysteine and glycine, it hydrolyzed by the dipeptidase to the glycine and cysteine. So, all these remaining 2 amino acids are now in free form. What happens to this gamma glutamyl amino acid? Gamma glutamyl amino acid, it is hydrolyzed by the enzyme gamma glutamyl cyclotransferase. What this enzyme will do? It will break down this peptide bond and this amino acid will be released. So, amino acid which was initially in the lumen, it is now transported inside the cell. And what happens to this gamma glutamate? Gamma glutamate, it undergoes cyclization and get converted to 5-oxoproline. This 5-oxoproline then acted upon by one more enzyme, that is 5-oxoprolinase. And this enzyme, it requires ATP. So, ATP gets converted to the ADP and 5-oxoproline gets converted again back to gamma glutamate. So, now we have all 3 amino acids, gamma glutamate, cysteine and glycine, all 3 are in the free form, and again it has to synthesize this glutathione. So, first this gamma glutamate, it binds with this cysteine. So, cysteine basically recycles, and this binding is carried out by the gamma glutamyl cysteine synthesis. So, enzyme is gamma glutamyl cysteine synthetase. This process also requires ATP. So, ATP hydrolyzed to the ADP, and so gamma glutamate and cysteine both binds by the peptide bond. So, gamma glutamate and cysteine, this dipeptide we get. Then this dipeptide, it binds with this third amino acid, that is glycine. Again this process requires ATP. So, ATP gets converted to ADP, and it also requires enzyme, that is glutathione synthetase, right. So, again glutathione gets synthesized. So, glutathione, it cycles continuously and meanwhile amino acids get reabsorbed. This process of Meister cycle for the absorption of amino acid, it is expensive one, right. For each amino acid absorption, see, we require this 1, 2 and 3 ATPs. So, 3 ATP are required for the absorption of 1 amino acid from the intestine. So, this is the expensive process. So, these are all different mechanisms for the absorption of end product of digestion from the intestine. And along with this absorption process, we had also understood many clinical aspects, including various disease and concept of super ORS. I hope that all the concepts are clear.